Hi guys, following on from my last video, an introduction to photosynthesis, I'm going to be talking about the light-dependent reaction. I've done some drawings already just to save time because there's quite a lot of things to go through, but let's see how we get on. So the light-dependent reaction, as the name suggests, requires the use of light. It's the first stage of photosynthesis, and remember that photosynthesis basically converts light into glucose. It converts light into glucose using the formula CO2, 6 CO2, we'll do the proper one, plus 6 water molecules, plus energy, which is in the form of light. And that creates our glucose molecule, C6H12O6, and our 6 molecules of oxygen. Okay, so we have our CO2 and our water and energy go in, and our glucose molecule comes out with a byproduct of oxygen. Okay, so hopefully you remember that. Another quick overview is where it all takes place. You have the chloroplast, and inside the chloroplast, remember this is a photosynthetic organelle within a plant cell. You have a double membrane, you have a stack called grana or a granum, and the stack is made of these little round things called thylakoids. These are very important in the light dependent reaction. You also have this area here, which is like a fluid substance called the stroma. You will see the stroma and the thylakoid membranes within this. When I talk about the membrane, make sure you realize it's not here, but it's the thylakoid membrane. Now, what we said, just talking about the light dependent reaction, which is the first stage of photosynthesis, one of our inputs, water, goes in, and so does light, our light, and out of this comes our oxygen. Okay, so one of the products of the light dependent reaction is oxygen, and there's another two products though which are used in the next stage of photosynthesis, and they leave here and go into the stroma where the light independent reaction takes place, the Calvin cycle. So one of them products is ATP, and the other product is reduced NADP. Remember, sometimes you'll see reduced NADP, and sometimes it can be written NADPH. Okay, it's the same thing. So hopefully that's been a bit of revision from the last video. Now we need to start talking about the parts of what's going on. So what we're going to do here, we're going to zoom in to this area here. Okay, and like I promised, I've already done a bit. Now, this is the thylakoid membrane. Not the membrane of the chloroplast, the thylakoid, the little discs. Here is the stroma, and this is inside the thylakoid. I hope that makes sense. It's important that you get that. And what we have within the membrane is 4 little proteins. 3 bigger than the other. So I'm just going to name them for you. The first set of proteins I'm going to talk about are the chlorophyll. Okay, and that's these 2 blue ones here. They're not blue really, it's just to help show you that they're different. Chlorophyll. And there's 2 types, okay, and they make photosystems. And the first one that you see is called photosystem 2. And the Second one is called photosystem 1. The reason this is called photosystem 1 and not the other way around is that this was the first one discovered. So when you write this down, make sure that photosystem 2 comes first. Both of these are chlorophyll, but they detect light at different wavelengths. This one detects light, photosystem 1 detects light at 700 nanometers, and photosystem 2 at 680 nanometers. That's just the wavelength of light. What we also have here is an electron carrier. There's quite a lot to take in, that's why I'd recommend that you watch this video a couple of times. So this electron carrier, it carries electrons across here. And the last thing you get, which I'll just have to move over here, is your ATP synthase. Now this is a protein that sticks ADP plus P together to produce ATP. Remember, this is one of the products of the light-dependent reaction. And basically what goes on in all this is electrons get placed in photosystem 2. We'll just say I'll say where they're coming from in a minute. Photosystem 2, they get energy from the light and then they move around this thing, jump in here, move around a bit more, some stuff happens, and it's all about the electrons. And because it's all about the electrons, this whole system, that's a wobbly line, is called an electron transport chain, because electrons go down it or across it, and then they make the products that's required. So you've got the stroma, the thylakoid membrane, inside the thylakoid, and you have photosystem 2, photosystem 1, electron carrier in the middle, and ATP synthase. Now these are all the working parts. Remember, we're talking about this little bit here. And then basically, stuff is made from here. Some of it's oxygen, and the other stuff goes over to the next stage, the light-independent reaction. This stage requires light. So go over to my other drawing, because actually it's a lot to draw on one picture. Now, all these steps that I'm going to talk about all happen pretty much at the same time. So we have to pick a stage. In the AQA syllabus, they talk about light excites electrons in the chlorophyll first. I'm going to talk about that second, because I think you need to know where the electron comes from. So we're going to do that first. There'll be a couple of main terminology you need to get, but I'll explain that as we go along. It's quite easy to explain like this in this manner. So the first thing we're going to talk about is the first stage. It's electron transport chain. We need to get some electrons. One of the inputs I said earlier was water, and that is where the electrons are going to come from. Now the water comes from the roots, and it is split by light. So light comes in, and it splits this water molecule, and this is called photolysis. Lysis, the splitting photon light. Okay, so it's the first stage. Light comes in and splits the water. This is called photolysis. And what happens is it takes, I'll use blue, it takes the electron, I'll use black then, takes the electron, and this goes into photosystem 2. Remember, hydrogen has one electron, so now that's not hydrogen anymore. So once photolysis takes place, you end up with 2 protons. These are protons, like I said, because hydrogen has one electron The electron's taken away, so now it's just protons. So we end up with 2 protons, and we end up with half an oxygen molecule. Okay. So this is where the oxygen is given off. Enough of these water molecules, you'll end up with lots of oxygen. So we've got our water coming in, our oxygen coming out. It's split by the light in a process called photolysis, which drops off the electron. That's the first stage. What happens next to this electron, now we're going to follow the journey of this electron, is the light excites the electron, which basically gives it more energy, and it's normally drawn like this, and it gives the electron more energy. Then the energy from the electron drops, and as it drops, the electron comes into the electron carrier, which is going to take it over to photosystem 1. Remember, the second one is photosystem 1. Very confusing. As this transfers and this energy drops, what happens is the thylakoid membrane allows more protons that are on the outside in the stroma to travel through the membrane, and what you get as a result is a build-up of protons, or hydrogen ions, are the same thing. Okay? So as the electron moves down the electron transport chain through the electron carrier, more of these come through here, and you end up with a bigger build-up inside the thylakoid itself. Now that makes a gradient, and what this gradient wants to do now is balance itself out. There's more of these fellas in here than there is out there, and that's a problem. So it's always going to try to balance out. The only place they can do that is by going through a little escape route, and the only escape route within this whole membrane is inside the ATP synthase that I spoke about earlier. That's meant to be a tunnel. And what happens, it's the only way it can go through, so the hydrogen ions will go through here and fill back up outside here. But as they do, ATP synthase uses a mechanism which basically, these hydrogen ions going through here, these protons going through, make ATP synthase work, and what its job does is you get your ADP plus P. As this goes through here, this phosphate is stuck together with the rest of the ADP, ADP adenosine diphosphate. So there's 2 phosphates here, one there, and it becomes ATP, adenosine triphosphate. That's the second product that we talk about. Talk about oxygen and ATP. This process is, I'm going to have to find some room for this because it's quite a long word. This process is known as photophosphorylation. Make sure I get it right. A long word. Phosphorylation. Which means a phosphate is being added using light. Photolysis, the splitting of water with light, and photophosphorylation, the adding of a phosphate. So now we've got, using light, now we've got our ATP. There's one more product I said earlier. Let's find where it's gone. So we've done our O2, we've got our ATP. Now we just need to get our reduced NADP. And our reduced NADP is an electron carrier. It carries electrons to the next stage. What happens is we go back to the journey of our electron, and our electron goes into photosystem 1. and the electron again gets hit by light. I'll zap it in between here. Gets hit between light, and the electron goes to another high energy state. But this time it doesn't come back down an energy gradient, it gets attached to an incoming NADP. So NADP is coming in, again from the other stages are recycled, and the electron jumps on here, and because the electron gets added to this, if you remember oil rig, this now becomes reduced NADP, because adding an electron reduces it. Reduction is gain. So now we have our reduced NADP. If some of these things like the biochemistry of the ATP and the ADP plus P and the NADP stuff is confusing, just watch the video that I've made, and that hopefully will make a bit more sense. And that is pretty much what goes on. So you have these three systems, you have these three things going on. Light splitting water, which creates the oxygen. You have the journey of this electron, and as it loses its energy, it allows the protons to come in. And you have, as they go through ATP synthase, ATP is created, and then when the electron gets over to photosystem 1, gains more energy from the light, and it is added to NADP. Not as complicated when you see it like this, it's just trying to remember it. This whole system like this is called Noncyclic Phosphorylation. Noncyclic phosphorylation, which means that this electron doesn't get reused. It starts off at the water, goes down the electron transport chain, and then gets dropped off on the NADP and goes off somewhere to the next stage, which we'll go to in another video. There is another type that happens called cyclic phosphorylation, and basically that only uses photosystem 1, and the electron gets recycled, keeps going round and round and round. It doesn't get dropped off.